I just left this slide up so that anybody who wants to join our campaign, this is a web address, go there and sign on. So what I'm going to do today is to tell you not just about the serendipity or luck or whatever it was that enabled us to discover splicing, but to tell you how luck played an enormous part in my life. And I think for many people, when you look back on the things that happened to you during your life, things were not always planned in the way that you thought they might be. And things have a habit of taking a different path than you least suspected. And that has been incredibly true for me. Now, I started off, I mentioned at the press conference, or whatever you call it, at the dialogue that we had, that for me, I learned something incredibly important. When I was probably about 15 years old, I was really good at this game of snooker. I was the Western England champion at that age. I was one day playing in a tournament, and I made a lucky shot. I tried to do one thing, it didn't work, but I potted the ball anyway and I could continue. And then I just messed up the next shot. And at the end of that time, an old gentleman came up to me. He was, I think, in 80 or so. He was a very good billiards player. And he said, you know, you are never going to succeed in life unless you take advantage of the luck when it comes. The worst thing you can do is to have a lucky break and then feel guilty about it because the other person didn't have it. If you don't take advantage of it, you're a fool. And I think that that is really very good advice. Everybody has luck. I don't care who you are, how old you are, where you are, you always have luck. And if you don't take advantage of it, then you're a fool. So if you remember nothing else from this talk, then I would say this is a good thing to do. And you will see that during the course of my life, I've had the most unbelievable luck, including one piece of luck that I will tell you about right at the end, that you may not believe. So I started off at high school, went to university. I went to the University of Sheffield. I went there because I couldn't get into Oxford or Cambridge. I failed physics, of all things, when I had to do A-levels the first time around. And so I ended up being too old to apply to go back to Oxford or Cambridge. And so I applied to a number of universities around in Britain. This one accepted me, and another one was the University of Southampton. But at the time, I was incredibly interested in chemistry. I got interested in chemistry by making fireworks and all sorts of things. And I was very lucky there to have two parents who were uneducated. They both left school at 14. And when I was doing chemistry experiments at home, they had no idea what I was doing. And so I was free to do all sorts of interesting things, including making explosives, fireworks, and so on. And my father, who was unbelievably supportive, would go down to the local pharmacist and get me chemicals that, you know, nowadays you would never be allowed to buy. He managed to get me sodium and potassium and all sorts of things. So I went to Sheffield to study chemistry. I would mention one thing, that I'd started off life loving mathematics. I really thought if you'd asked me when I was about 10, 12, 15, mathematics was the thing that I was going to do. But I got converted by chemistry because I liked the experiments. So I went to Sheffield, had a very good education there, and I met this gentleman, Kazu Kurosawa. When I started to do my PhD, I was put in a lab, and Kazu, he's older now. This is a picture I took of him just last week in Tokyo, where we met for the first time. It was 50 years since I'd seen him, and I met with him and we talked. He played the most incredible part in my life because as a postdoc, he would suggest to me and tell me what experiments I should do. I'd been given a project, I got started on it, and he was one of these very rare people who not only can tell you what to do experimentally, but at the end of it, you understood why you were doing it. And you could then go and you knew why you had done this experiment, and it usually worked. And within a year, I had all the results I needed for my PhD. So can you imagine that? 1 year, I could have written my thesis. But in England, you're not allowed to do that. And so I had 2 years to find something else to do as a graduate student. And so I started off thinking about what I was going to do with my chemistry later on, when eventually I left the university. And I started to go to lectures in other subjects. I listened to visitors who were coming in. But I spent a lot of time in the library. I've always been a very avid reader. I love to go and read about all sorts of things. And I went browsing around in the library, and I came across something called the Journal of Molecular Biology. And since I was a chemist and working with molecules, and I'd actually been doing natural product chemistry, it seemed to me maybe there was something in this biology stuff that sounded okay, and molecular sounded pretty good. And during the course of that, I went to look for books that might be appropriate for molecular biology. And I read this book called The Thread of Life by John Kendrick. Now, John had won the Nobel Prize for the discovery for doing some X-ray crystallography work on myoglobin. And when I finished reading that book, I knew I wanted to be a molecular biologist. It was like night and day. I started off reading it, it sort of sounded interesting, didn't know what I was going to do with my life. By the time I finished that one book, it's not a big book, I knew this was what I wanted to do with my life. And I would encourage all of you to go and read about stuff that is outside of your normal subject. Okay? You never know when you're going to have the same experience of suddenly reading about something and saying, wow, that looks interesting. I'm going to pursue that. And you may well discover that you can become passionate about something that is totally different from anything that you have thought of. And so that was really important. The other thing that Kazoo taught me was how to play Go. I have a Go board down here. And again, he was such a good teacher that after I left Sheffield, I really played a lot of Go. And it wasn't until about 12 years later that I met anybody who was not Japanese who could beat me at Go. And I ended up, there was a German postdoc who happened to be at Cold Spring Harbor, and we started playing, and he turned out to have spent most of his life playing Go, and he was very good at it. Unfortunately, I've sort of stopped now. I've not played Go for a long, long time. But it's a wonderful game. I don't know if any of you like games, but you should think Go is a good game if you don't play it. Much more complicated than chess, more difficult to learn, and yet incredibly simple in terms of the rules. They're just 2 or 3 rules and you're away. So the thread of life was important. I wanted to be a molecular biologist, and so when it came time to look for a postdoc position, I applied to labs that were doing biological chemistry. I thought, I can't really go and try and apply to be a molecular biologist because all my training was in chemistry. And I thought maybe if I go into a lab where people are doing sort of biology of one sort or another, biochemistry, this kind of thing, it might be good. I applied to a lot of universities. Only one person accepted me, and that was this man, Jack Strominger. And he accepted me for a very interesting reason. Again, very, very serendipitous. It turned out that he liked to hire postdocs who came from the country where a previous postdoc had done well. So if he had an Australian came, very good Australian came to his lab, and the next 3 or 4 people from Australia who applied to work for him, he accepted and took them. Until he got one who was bad, and then he didn't do that anymore, and so then he picked another country. And I was lucky that he had an English postdoc who had done really well with him, and I was the next one to apply, and sure enough, he gave me the job, made me an offer. But even better than that was that he was at Wisconsin, University of Wisconsin at the time, and at Wisconsin, he had done really pretty well. He had done all of the primary work to understand how bacteria make their cell wall. And this had drawn him to the attention of people at Harvard. And instead of going to Wisconsin, which I thought I was going to, about 2 months before I was scheduled to go, sent me a letter and he said, can you delay for 2 months because you're going to come to Harvard, not to Wisconsin. And so I ended up going to Harvard instead. And I had 4 years as a postdoc at Harvard, and those were unbelievable years. I got so much done. I learned so much. I had a fantastic time. And all because of the fact that Jack had just suddenly gotten this job at Harvard. And something else had happened, was that he had a reputation as being a slave driver. He had a reputation for really treating students, postdocs, very harshly. You had to work hard. But it turned out his lifelong ambition was to get to Harvard. And so here he was. He arrived just a month before I arrived. I get there, and here's a man who suddenly achieved his lifelong ambition, and he changed completely. He let me do whatever I want. He didn't tell me do this, do that, do the other. And he was everything except a hard slave driver. I also got put on a project that was finishing up something that actually a previous Australian had done. And he, I thought, had not gone about the process of solving this problem, which was to sequence a transfer RNA, in the right way. And so I read about it a little bit, and I've got to tell you, I had a hard time at Harvard. It took me about a year to be able even to talk the language of the lab I was in. But nevertheless, I did manage that. I decided I was going to sequence this tRNA using a new technique that had been developed by Fred Sanger, one of the few people who won 2 Nobel Prizes. I went over to his lab in Cambridge, England that is, and learned how to do p32 sequencing, radioactive sequencing. Went over there, learned, I spent 6 weeks, I wrote down everything that I possibly could. I came back to Harvard, set up a lab, a sort of a smaller version of the MRC lab in Cambridge that Fred had, and we started sequencing. And it turned out I was the first lab in the Boston area to actually be doing Fred Sanger's p32 sequencing. And so everybody came to me to learn how to do it. And here I am, just a postdoc. I've switched fields, just getting going. Wally Gilbert came to my lab to learn how to do it, and Alan Maxim. Mark Patan, very, very much. And it is a great way to make progress in science. So always think when you can, whenever it's possible, help people. Now, one of the things that happened to me while I was at Harvard was I went to a lecture at Harvard Medical School by the man on the right here, Dan Nathans. And Dan Nathans had been part of a group of people that had found and worked out what might be useful for this group of proteins called restriction enzymes. These were enzymes that would cut DNA into very precise pieces. And one of the things that had happened in terms of sequencing RNA was that really you would like to be able to sequence DNA, but there were just no methods available. And no methods had been developed. And the reason was that if you want to develop a new method to do something, you have to have something to practice on. And all of the DNA molecules were huge. But RNA was small. tRNA is 75 nucleotides long, very small, and so it's easy to work out methodology. But if you've got something where the smallest DNA we knew about at the time was about 5,000 nucleotides long, you didn't even know how long it was, this is a really tough area in order to work out methodology. And I thought these restriction enzymes were able to cut DNA into pieces, and maybe this would allow me to develop methods for DNA sequencing. And so when I got offered a job by Jim Watson to go down to Cold Spring Harbor Laboratory, and again, this was an incredible piece of luck in my life, for some reason, one of the people, Mark Patashny, who I taught how to sequence RNA, had been talking to Jim Watson. And so one day, Mark comes up to me and he says, "Oh, Jim Watson wants to meet you. He's going to come and see you and offer you a job at Cold Spring Harbor." Right? And so I wait a week and a couple of weeks and nothing. And so then Mark comes up to me again and he says, "Oh, you better go and introduce yourself to Jim because he doesn't know who you are." I go, I knock on the door, I walk into Jim Watson's office, and within 10 minutes I left with an offer of a job at Cold Spring Harbor in my hand. And I was just about to leave, and he said, "Oh, by the way, you probably should come down and visit us, but you've got the job, don't worry, you've got the job." And this is, I think, pretty typical of Jim Watson. He sort of made up his mind. And he'd made up his mind because of Mark Patashny. Mark had told him that he thought I was okay. Jim had a specific problem in mind. He wanted SV40 DNA sequenced. And the way that people were doing sequencing in those days on DNA was by copying it into RNA. So I get down to Cold Spring Harbor and I discover there are already 2 groups sequencing SV40 DNA. And so it seems stupid. You know, why would I want to do it too? There are already 2 other groups doing it. I want to do something different. And so I thought, well, what I'll do is I'll tell Jim that I'm sequencing SV40 DNA, but in fact I'll try to work out some new methods for sequencing DNA that are all based on restriction enzymes. And so I made some of these restriction enzymes that were already known. There were 6 known at the time. I made those, and then we started looking to see if there were others. Because it turned out one of the ones we made was actually a mixture of 2, and then another one turned out to be a mixture of 2, and before we knew it, we isolated a whole slew of these restriction enzymes. Now I couldn't figure out how to get these used in a form that would be useful for sequencing DNA, but it seemed to me these were so useful all by themselves that this would be something worth focusing on. And so much of my scientific career has ended up focusing on restriction enzymes, things that they catalyze and using them for various things. And these have really been my bread and butter project, if you like. This is what I've done throughout my scientific career. And it's been fantastic. Among other things, one of the things I'd suggested to Jim Watson after we had about 30 of these, and everybody wanted them, that we should start a company at Cold Spring Harbor and use the profits to support the science at Cold Spring Harbor, to which Jim said, no, you can't make money at this, I have no interest in this, plus it's really dirty to be associated with companies. And he didn't want to do it, he didn't like this at all. Unfortunately, I found some other person who did. New England Biolabs, the company where I work, ended up being the beneficiary of all of these enzymes that were discovered in my lab at Cold Spring Harbor. New England Biolabs does probably $140, $150 million a year in business. And much of this income, the profits, we put back into research at Biolabs. If we'd done it at Cold Spring Harbor, we could have put it all back into research. So, you know, when I left Cold Spring Harbor, about 15 years after this, Jim did admit that this was a mistake. Perhaps he should have listened to me and done this. It wasn't the only mistake that he made. Anyway, so this was Cold Spring Harbor. So I went to Cold Spring Harbor. Shortly after I'd gotten there, I got an application from a postdoc from this young man, Richard Gelinas, not so young anymore, but nevertheless. And I had met when he was a student at Harvard and I was a postdoc, and we got on rather well together. And so he applied and said, could he come and be a postdoc at Cold Spring Harbor? He was working on a completely different project. He was working in the lab of Potus Kafatos. And so I said yes, because it seemed it would be fun to have him around. He came down and we started to figure out what might be a decent project for him to work on. And so we, at the time, knew what bacterial promoters look like. These are the sequences where you start making RNA from DNA. And everybody, and I mean everybody, thought that bacterial promoters, eukaryotic promoters, all promoters were going to be the same, because they thought the fundamental processes of DNA makes RNA makes protein was universal. I think it was Monod or Jacob who, in a famous quote, said, well, you know, if you understand bacteria, you understand elephants. That, you know, life was basically all the same. They thought, after they'd done the genetic code and everything else, that they had solved the basic fundamental mystery of life, and the rest of it was just details to be mocked up. So I thought, well, we're actually in a position we can use our restriction enzymes in order to tackle this problem. And so Richard thought this was a good idea, just to see, let's see, you know, people think the promoters are the same, let's just test it and see. And so we got started doing this. Let me just go briefly and show you what we had in mind here. Can it work? No, maybe not. Okay. But anyway, you can see up here, this is what a typical gene looks like in a bacterium. The red area is the actual gene. This is the region that is going to code for the protein. You start making RNA at this point here, go all the way through, stop at the far end, and then you take the RNA that is produced, and it also has on it a start point and a stop point for the gene. Then you send it to the ribosome and it makes protein. So very simple, straightforward thing. And so what we wanted to do, get the next slide, I'll try to do this pretty schematically. So in a bacterial DNA, you've got loads of genes, and they're all one after another. There are little spaces here in between. These are the signals that tell the RNA polymerase where it should start and stop making RNA. And then you come down and you get all the way down and make everything in the cell. In adenovirus, which was a small human virus, we thought it would be good to use this to try to characterize a promoter. And we did it for several reasons. One is that it was growing in the lab. It's a very nice virus to work with in the lab. It grows in human cells. It's not dangerous, although most people, when they're very young, when you're 1 or 2 years old, you get an infection with adenovirus, you get symptoms of a mild cold, mom thinks you had a cold, treats you nicely for a day or two, it clears up, and then you have antibodies against adenovirus, you never ever get another infection. And people in another lab at Cold Spring Harbor had started to work out where the genes were, and they'd been using our restriction enzymes, and we worked with them a little bit, to map where the genes were. And so we knew that there were early genes over here, some late genes over there. mRNA would be made, we felt, the same way that you made RNA in bacteria, and then you would make the proteins, hexon, penton, fiber. These are the proteins that surround the virus within the virus particle when it's going to come out. So the concept of our experiment was as follows. Here's a gene, here's the RNA transcript that is made. Just upstream of it would be the promoter, and it would be just upstream of the start point. So if we could get a little bit of DNA sequence in this area, and in particular, to find the little bit that was at the 5' end of the RNA, which is what I knew how to do, because this is what I've been doing at Harvard, we knew how to get at this, and then we could do a bit of sequence up here, and we could find what the sequence of the promoter was. Now, the adenovirus genome looks like this. This is an actual map that we had, or that other people had developed back in about 1975, 76. There were early and late transcripts. So all the late protein transcripts were all going from left to right up on top. Down here were some early transcripts. And the important thing is adenovirus is a linear genome. And so we knew that there were early genes that came in from the end of the genome over here, and an early gene that came in over here. And so this meant that if we could get the sequence from the very end of the adenovirus genome to the point where the first transcript was being made, it would have to have the promoter in it. And we had independently been working on sequencing the very ends of adenovirus, and so we were already well underway. So we thought this was a good project to go on. Now, messenger RNAs have a very interesting structure. They all start with a triphosphate, and this comes from the ATP or GTP that is being used to get things started. And in eukaryotes, at the 3' end, they have this long poly-A track. Now, there was an enzyme that we used to sequence RNA called ribonuclease T1, and it cuts after every G residue. So wherever there is a G residue in the RNA, it will cut, and so you end up with a series of oligonucleotides, all of which terminate in G. But you'll notice the one that was at the 5' end has this funny triphosphate structure on it. The one that comes from the 3' end has poly-A on it. And it turns out it's quite easy to begin to separate some of these. So the AAA is easy to get rid of. You just take a long oligo-DT, something's got all T residues, hybridize it, and then that all disappears. That was well known how to do that. And so what we were interested in was how you differentiate these internal nucleotides from the external one. And we were busy thinking about how to do this when another discovery was made. And this just shows you how important it is that you sort of keep track of the literature, see what other people are doing, because it was discovered that in eukaryotic messenger RNAs, they don't have a triphosphate at the 5' end usually. Rather, this triphosphate, which would normally lead straight into the RNA, has this complicated cap structure put on it. And this was fantastic because all of a sudden, it turns out that you have a structure at the 5' end telling you which is going to be this 5' nucleotide that starts everything. and it's got a cis diol on it. And so there are chemical ways in which you can isolate this. And so we developed a new method for separating the capped oligonucleotides, the things that were right at the start of the messages, and then getting rid of everything else. And so when we did that on late mRNAs, we tried it on earlies, but we couldn't get enough RNA, so we ended up looking at late RNAs. When we did that, what we discovered, what we were expecting, was we knew there were about 8 or 10 different genes that were all expressed late. We thought each one would have a separate message, and that the sequence at the 5' end at the start of the RNA would be different on every one. And so we thought we can get a very nice little feel. It's sort of like doing a microarray. We thought, you're going to get 10 different oligonucleotides. You can spread them out. There was a two-dimensional way to spread them out. And we would see 10 oligonucleotides, and they would all be the ends of the messenger RNA. We thought this would be great, because then we could look what happens during an adenovirus infection and monitor when the RNAs were being made, when they were not being made. We thought it was terrific. So Richard Gelinas did the experiment. Okay, so this is the way we did it. And this was the result. Okay, so when we looked at these things, early during these 2 to 24 hours after infection, there were 2 spots here. Wait longer, there's only 1. It turns out the 2 spots here are related to one another. They just differ according to whether the phosphate is cyclized or not. But where were the 10? We were expecting 10. And so, of course, being a good supervisor, I suggested to Richard, well, he should go back and repeat the experiment. He must have screwed up along the way. And so he goes back and is very careful, does the experiment all over again, gets exactly the same result. So I said, Richard, look, sit down, I will do the experiment, I'll show you how to do it, and we'll see the 10 spots. So I did the experiment, and lo and behold, I got the same result. And so at that point, I started to believe it. If you do it yourself, you believe it. If it's a student, you never believe it the first time. So here we have a conundrum. Here we know there are 8 or 10 genes. We were expecting 8 or 10 messenger RNAs, and yet they all have exactly the same sequence at the 5' end. So what is going on? How can you explain this? This was a paper that described that. This was published about 2 months after we actually knew what the answer was, but of course this paper had gone in considerably earlier. So we started to look at this capped oligonucleotide, and the obvious thing to do was to isolate individual messages, if we possibly could, and check that each one really did have this same sequence at the end. So we did that. We hybridized the RNAs to different pieces of DNA along the adenovirus genome. They all had the cap. A man called Dan Klessig, who was a student working in an adjacent lab, was also working along with us on this project, and he looked at 2 absolutely pure RNAs, hexon and fiber, and that too had this cap on. However, if you took the RNA and hybridized it to the coding region, the gene, and then treated it very gently with ribonuclease, you lost the cap. The cap disappeared. The mRNA was there, but the cap disappeared. And so that was a little odd. What it said was that the cap structure, this undeckered nucleotide, probably wasn't being made from the sequence that was immediately adjacent to the gene. And I'll show you schematically what that means in a moment. We also found, thanks to this Indian lady, Saeeda Zain, who worked in my lab as a postdoc at the time, she was looking at adenovirus SV40 hybrid viruses where some of the genes had been knocked out. And in this particular case, she showed explicitly that this capped oligonucleotide could not be coded right next to the gene. And then we found that the early RNAs have a totally different terminal oligonucleotide, but that's a separate story. So this is schematically what we're observing. We take a DNA strand that is a part of a gene and coding for the messenger RNA from which that RNA and protein has to be made. When we do the hybridization, we were finding that this capped oligonucleotide at the 5' end was not coded there. just as this poly A that is at the 3' end is also not coded. We know that the poly A is not coded because there's an enzyme that adds it at the 3' end of the RNA to help it from getting too degraded. But what's going on here? Where is this coming from? Well, the answer was we didn't know. And so we started to think, well, how might we find out where it's coming from? Unfortunately, a technique had been developed out in Norman Davidson's lab at Caltech, in which you could make hybrids, in which you take a messenger RNA and you hybridize it to a double-stranded DNA that you've partially melted, and you can form these things, which we call R loops. And so it turns out because a DNA-RNA hybrid is more stable than a DNA hybrid, that you can actually form these nice big loops in the DNA if you have a pure messenger RNA to put down. Another man had shown that when you do this in a normal RNA, and you can add oligo-DT to this, you can actually label this and find out the direction, which part of the loop has the 3' end on it. We thought, well, if we could find the DNA that would code for this and would hybridize to this, we could do the same thing. We could make an electron microscopic label. This is the so-called R-loop experiment. We made the R-loops. I had a guess as to what I thought was the DNA fragment that was going to code for this piece. Turned out to be totally wrong, as always. You know, every time you come up with a great experiment, it never quite works out the way you want. But nevertheless, even though it was wrong, it gave us the answer that we needed. And this is where Tom Broker and Louise Chow came in. Now, Richard and I had come up with this experiment one Saturday morning. We've been in the habit of sort of getting into the lab and talking on a Saturday morning why the last week's experiments hadn't really worked the way we wanted them to. And so we would be sitting down and we'd have some coffee and chat. And at the time I used to smoke, so I'd be smoking a cigarette or two. And we would go through and think, how can we do a better job than we did last week? And so Richard was expounding this incredibly complicated experiment to me, and my mind was wandering, and that's when I came up with the R-loop experiment. The only problem with that was that neither Richard nor myself were electron microscopists. But fortunately, just down the hall were two electron microscopists, Tom Broker and Louise Chow. Very talented, Louise in particular. And she was an incredibly, and still is, a very talented electron microscopist. And so we went down, and they fortunately were also working on a Saturday morning. And so we talked to them and said, you know, we have this idea for an experiment, would you do it for us? And so they looked and they said, well, you know, it's never quite been done, but yeah, I guess if you make the reagents, we could do it. And so Richard went off, made the reagents that we needed to do this experiment. Tuesday morning, we went down to Tom and Louise and gave them the reagents, and the very first molecule that they looked at looked exactly like the one that I'd drawn on the board in the previous schematic. So here was the R-loop. There was the R-loop with the probe on it. The probe DNA that we had selected exactly matched and formed this tail that you could see in the electron microscope. The only problem with it was that instead of forming a nice structure, which I thought it was going to do, it had formed this loop structure. And we were lucky that the piece of DNA we'd chosen, and I'd chosen it not because of these sequences, but actually because of a sequence up here that I was expecting to be the source of the oligonucleotide, and it turned out that it contained two stretches of RNA that were attached at the 5' end of the main body of the messenger RNA. And so this was unbelievably exciting. I mean, to do an experiment with an electron microscope where you look at one molecule and get a result that is unexpected, is different. Normally, you have a lot of biochemistry to do, for the simple reason that electron microscopy looks at one molecule at a time. And when I take a mixture of molecules, you know, there are thousands upon thousands of them, so you want to look at a lot and then do some bio. We'd already done all the biochemistry. And so at this point, we more or less knew that we had discovered split genes. And I can tell you, by the afternoon, when we'd gone around and told everybody who would listen to us at Cold Spring Harbor about this, they were all talking about Nobel Prizes. This was in 1977. Well, you know, a bit early at that time to be doing that, but anyway, we got the result we were hoping for. And what that meant was that the split or the late adeno genes were split into pieces. So when you were making messenger RNA, you made a piece from here, piece from here, piece from here, piece from here. It was made as a single transcript, and then that transcript was cut and re-spliced back together in order to make the final mRNA, which didn't have these sequences in between, which we call introns. And the way you can think about this is when a Hollywood director is making a movie, what he does, he takes a series of clips of film and then puts them together by cutting and splicing so that at the end of it, it looks as though it was just one scene shot all the way through. And that's exactly the way in which eukaryotic cells make their messenger. They make a long transcript, cut it, and splice it to get a messenger RNA so that the messenger RNA actually looks like a bacterial messenger RNA by the time it gets to the ribosome. So this was incredibly exciting. And of course, the reason I showed you schematics is because this is what the real data looks like. You can see all these little black things. There's actually a loop here, a loop here. Schematically, it's colored. You can see it a bit. But the actual electron microscopy and picture is very complicated, not easy to show in this sort of audience. So that's why I chose to use the schematic. This was the paper that we published about this. It was called An Amazing Arrangement of the 5' end of messenger RNAs. When we sent it off for review and got the reviews back, we had 3 reviewers, and they each said, well, they didn't think it was appropriate to use the word amazing in a scientific article. You know, it's not something you put in the title of a paper. And so the editor had also written and said, well, you should change it, find another word. And so I called him up. I knew him a little bit. I called him up, Ben Lewin, and we started talking. And so, you know, I said, when you first heard about this, what did you think? And he said, oh, it's amazing. I said, I rest my case. So this, as far as I know, is the very first paper ever to use the word amazing in the title of the paper. Now, time goes on. In 1993, I eventually got the call saying that I had won the Nobel Prize. One of the things you have to do, you've got to go to Stockholm and talk about it. Now, I had not worked in this field for about 10 or 12 years, and so I was wondering, you know, what am I going to talk about when I give my Nobel lecture? And at this point, I thought, well, you know, it's just going to be boring to talk about the original discovery, because Phil Sharp, who I shared the prize with, had gone on and worked in this field and done loads of work, and I'd really done nothing. And so just before I had to decide what I was going to talk about, we got this result. And this is the result of the DNA methyltransferase binding to DNA. And what this enzyme does when it binds to DNA, here's the double helix coming down here, but this base, which is the one that the enzyme is going to put a methyl group on, is flipped right out of the helix into the active site in the protein. And we call this base flipping. This was the very first example of base flipping. We now know it's incredibly common. In fact, we think it likely that this is the way in general that proteins start to open up DNA if they're going to either replicate it or they're going to make messenger RNA, and there's some reasonable evidence for this now. And so I use this as the basis for my talk in Stockholm. And you know, if you've got to give a talk about a new discovery somewhere, Stockholm's not a bad place to do it. But I'm still waiting for the follow-up from that. So, what I just want to do while I finish is to recap the importance of luck and serendipity. It has been incredible in my life. So, the first one, of course, was the billiards story. You know, this old gentleman who really said, when you find something new, take advantage of it. And so I've always tried to do that whenever I could. I found this postdoc, you know, by chance, I ended up in the lab with probably the best mentor I could ever wish for. He was fantastic. He was just great. I ended up going to Harvard instead of Wisconsin. And then we started looking for promoters in adenovirus. We were expecting them to be pretty similar to bacterial promoters, maybe a little bit different in sequence, but we were not expecting to discover RNA splicing. So that turned out to be a really good move. I want to tell you about the biggest piece of luck that has happened to me in quite a long time. I call this taking an early plane. You probably all know what happened in New York City when the Twin Towers came down. The first plane that went into those towers was flying from Boston to LA. I was booked on that plane until 2 weeks before it took off. And the meeting that I was supposed to go to was scheduled and moved 1 day earlier. And so I ended up taking that plane 1 day earlier just because of this meeting change. Otherwise, I would not be here to tell you about this story. Luck, I've had so much luck in my life, I can't tell you. It's fantastic. And then finally, when I decided at Cold Spring Harbor to study bacterial restriction modification systems, this has been with me throughout my life. We've made a lot of interesting discoveries. I now do a lot of bioinformatics. I make discoveries all the time using these systems. I get excited every time I make one. It is fantastic. And most recently, we're working on methylation in bacteria. And there's a lot more methylation going on than we ever thought there would be. And I'm convinced there is something big there. My gut says here is a big discovery waiting to be made. Don't know what it is. It's one of these, you get a bunch of data and you start thinking about it. Not many other people have been thinking about it or working in this field, and I'm hoping I will figure out what it does before anybody else does. But I can tell you, if one of you discovered what it was doing and found some new phenomenon, which I think is going to be important, that would be just fine. I would like nothing better. So, studying bacterial RM systems for me has been fantastic. These are the people who were really important. Richard Gelinas, who did all of the biochemical work in my lab to characterize the undeconucleotide. Louise Chow and Tom Broker, who came along and did the electron microscopy at the end, and they were then able to follow through on that. And as I say, Louise is one of the best electron microscopists I've ever come across. Saida Zain was a postdoc who unfortunately is no longer with us, but she provided a very important part of the story that really proved for the first time using sequence that the capped oligonucleotide was not coded right next to the message. Dan Klessig purified these RNAs and showed that they too had the thing. And then finally Karen Otto, my administrative assistant, helped me tremendously with the slides. I thank all of you for your attention.